Mitochondrial myopathy is a rare genetic condition that happens when the mitochondria in our cell don't work properly. Mitochondria are often called the powerhouses of the cell because they generate energy in the form of ATP. This energy is what our body needs for movement, growth, and daily activities. Mitochondria have their own DNA, which is mitochondrial DNA, separate from the cell's nuclear DNA. Some proteins in the electron transport chain ETC are encoded by mitochondrial DNA, while others come from nuclear DNA. When mutations occurs in these genes, the structure of function of ETC proteins get disrupted.
In normal mitochondria, the electron transport chain efficiently produces ATP through oxidative phosphorylation. Under normal conditions, electrons from NADH and FADH2 are transferred through a series of protein complexes embedded in the inner mitochondrial membrane. This electron flow drives the active pumping of protons from the mitochondrial matrix into the intermembrane space, creating an electrochemical gradient. The resulting proton motive force provides the potential energy required by ATP synthase to convert ADP and inorganic phosphate into ATP, producing approximately 2 to 3 ATP molecules per NADH oxidase. However, in individuals with mitochondrial myopathy, ATP production is reduced due to mutations in one or more components of the ETC, commonly in complex 1, complex 2, complex 3, or complex 4. In this case, a mutation in complex 1 disrupts the oxidation of NADH, preventing the transfer of its high-energy electrons to ubiquinone, which is coenzyme Q. As a result, electrons transport through complex 1 is impaired and fewer protons are pumped into the intermembrane space. Although electrons from FADH2 can still enter the chain via complex 2 and continue through complex 3 and 4, the overall proton gradient is significantly reduced. This diminished proton gradient lowers the proton motive force, thereby decreasing the rate of ATP synthesis by ATP synthase. Additionally, the inability of NADH to be oxidized leads to its accumulation in the mitochondrial matrix. The increased NADH to NAD+ ratio shifts cellular metabolism towards anaerobic glycolysis, resulting in the conversion of pyruvate to lactate and ultimately causing lactic acidosis.
Mutations in the nuclear DNA are commonly inherited in an autosomal dominant gene, which means one mutated gene is enough to cause the disease, or in autosomal recessive gene, which means two mutated genes, one from each parent, can cause the disease.
In primary mitochondrial myopathy, muscle cells are unable to generate ATP, which result in muscle weakness and fatigue. Sometimes there may also be muscle pain, cramping, stiffness, or even paralysis of the muscle. Individuals typically develop exercise intolerance, which is a reduced ability for performing a physical activity. Unfortunately, there is currently no cure for mitochondrial myopathy, but treatment focuses on managing symptoms, preventing complications, and improve the patient's quality of life. Firstly, patients may require regular exercise to improve muscle function. Moreover, supplements called mitochondrial cocktail or mitococktail containing vitamins, creatine, antioxidant, and coenzyme Q10, which is a component of the electron transport chain, can be given to improve mitochondrial activity. Next, patients are often advised to avoid situations that worsen symptoms, such as this include extreme cold or heat, skipping meals, stress, and lack of sleep. Lastly, we must avoid mitochondrial toxin, being cautious with certain medications such as some antibiotics, anti-epileptic drugs like valproic acid, and anesthetic which can inhibit mitochondrial function. It is crucial for a person with mitochondrial myopathy to consult with a specialist such as a neurologist or geneticist experienced with mitochondrial disease to develop a personalized treatment plan.